Good morning, everyone, and welcome you all to this 31st day of the CSIR online training. Today, we are going to discuss about the important topics which we are not covered in the previous 30 day because that is very short duration. We refresh our topics, just go through the simple topics to overcome to understand the remaining part. I'm included some important topics in the uh, syllabus, okay, which I was missed in the past classes. Before I enter into the session, please let me know, am I audible? Can you see my slides? It's visible. Please let me know through chat box. Yeah, thank you, ma'am. Thank you. Okay, we will enter this session. In unit 1, that is the energy, that metabolism related pathway. In that, the bioenergetics is a very important one topic because in that bioenergetics we have covered many pathways, like oxidative pathway. So, the oxidative phosphorylation, coupled reactions, we discussed in a short manner, but in this topic, very important things are there. The thermodynamic, biochemical thermodynamics. And the thermodynamics means it depends on the parameters which involve in the pathway reactions. Each and every reaction, there is a parameters. The parameters means that is the temperature, heat, which how much energy utilized, how much energy released during the energy. All those things are comes under this thermodynamic concept. In this like here thermodynamic, it's a moving one. And bioenergetic, which compounds are moving, which are sharing between the atoms, between the cell to cell, and all those things are studied under this biochemical thermodynamics. It's a study of the energy changes. It's in the bioenergetic topics. Many researchers are working in India and also in abroad. And why we need to understand this concept means, for this bioenergetic concept, if you are utilizing less energy and producing the more energy in a cell. For example, if you particular species, that bacteria or anything or human, they are utilizing less energy and they are producing the more output. That kind of that is a very needed one in the latest period. So they developed this kind of researchers. On this, they are modifying the pathways involved in the energy metabolism. In this, three fundamental thermodynamic parameters are there: enthalpy, heat content of physical object, and derived from first law of it's derived from the first law of thermodynamics. These three parameters are here because in this enthalpy means it's a each and every step, the all atoms are bind with the bonding. That bonding may be the van der Waals, maybe caused by the van der Waals force. That binding may be caused by the hydrophobic interaction. That may be the polar interaction. They may be the charge based interaction, positive negative charge based interaction, or glycine interaction. Or because all pathways are activated by the proteins are made up of the amino acids are having their sharing hydrogen, nitrogen and carbon O atoms here. So they are making the bond with other protein, other molecule, with this protein and molecule interaction or protein interaction. Everything they are calculating the enthalpy. the H and O make a bond with the means that utilizing some energy. That energy is calculated for the is added for the this enthalpy. In the this enthalpy value is calculated, that is energy utilized, that calculated based on the each and every bondings, not only one particular bonding, each and every bondings utilizing some energy, they can calculate in the for calculating for the bioenergetic calculation. Okay, so heat content. For if there is energy utilized and the cells are getting processed, means that processed cell, that is a physical object, should be in heat mode. So as a heat content of how much heat is generated, that is the output, is a physical object. Then it's the first law of thermodynamics. This energy is shared between one cell to another cell. That they are communicating between each other, so the energy is transferred, and it's one energy is some energy utilized and some energy is released. In this in our human body, many systems are involved in the bioenergy transferring. Okay, the energy transducer, because as I mentioned, it's one continuous process. If the, for example, you are doing exercise, means your muscles are making that contractions are happen in a muscle, that contraction make a signal to individual cells, that individual cells are activated, then release the energy, that energy make the heat over the body, that heat system helps to release the sweat out. So there is a water removal in a cell, so the cells are the removed waters are released as a sweat. So if a body getting heat, that make a sweating. So all those things are sequential process. Like a normal sweating is different than the exercise. During the exercise you are getting sweating, it's all the sequential. So likewise, the how they transduce this signal. Okay, in every here, so I mentioned some example, and next is upcoming slides I am also include some examples. In this slide, how the membrane acts as an energy transducer. In this membrane, you can see the inner leaflet. We know the membrane having the inner polar, the inner the lipid bilayer. In that bilayer, they are having the integral membrane protein, interprotein, asymmetric chemical environment, means the cell inner surface having some chemical environment differ than the external environment, then only they can survive on that particular temperature, otherwise it can't survive on the particular temperature. So the chemical compositions, chemical environment in a cell should be different from the external of the cell. That if the medium, the cell internal part is contains some chemicals, that if you are adding that cell into one medium, means that medium should differ from the internal compositions of the cell. If there is a similar, means that won't grow, that cells are destroyed. Then outer leaves, this all those proteins, that membrane integral proteins are involved in the transduction process, and this polar compounds also in the transduction process. The glucose, when we are discussing the cell membrane, we studied these compounds are the sugar molecules. Like this, all those things are involved in the transduction. In the intracellular energy distribution, here transduction means it may communicate for outer or communicate for internal. Here I am included the intracellular, that is the internal energy, the energy distribution, how it energy distributed. Okay, first is three pathways: the phosphor transfer reaction, Then mitochondrial reaction, the glycolysis process. This things are uncontrolled condition because they are uncontrollably releasing the ATPs. There's a mitochondrial ATP source. In this, they are activating some pathway. For example, is ATP, it's activating the muscle contraction ion transport. How is activated that? Using the ATP utilized by the creatine kinase and adenylate kinase. These two enzymes are involved in the that activate that energy transduction, that activating and transduction for the muscle contraction and ion transport. Okay, and then the polysaccharide production. For polysaccharide production, they are utilizing this creatine kinase, adenylate kinase, and the nucleoside diphosphate kinase. This is the thing. And likewise, they having for 5 different energy transducers. That is the protein which helps to transduce the that transfer the energy. Then nucleoside monophosphate diphosphate kinase is different, and monophosphate kinase in the protein they are utilizing the diphosphate kinase, nucleoside diphosphate kinase. In lipid production, they are utilizing the nucleoside monosaccharide kinase and thymidine kinase, which involved in the RNA synthesis, DNA synthesis. The hexa kinase and means that adenine nucleotide translocator. In the DNA synthesis, there's 3 things are very important. Thymidine kinase, that is a hexokinase, then and adenine nucleotide translocator. These are the proteins, the enzymes which involves in the energy transduction of intracellular process. Then some extra other than that biological transduction, these are all the some compounds which involved in the that organelle cell transduction. That is the cell, that internal cell, how they communicate. Here the organ as organ to organ, the energy transducers, biological transducers are there, which are listed here. In this case, RS Odafism, it's helpful for transfer the energy transduction of retina, and chlorophyll, it's energy transfer compound of the biological transducer is a chloroplast. And the egg, that is the electric and nerve membranes, are the transducers. And actinomycin, it's involved as in the muscle cells, muscle cell energy transduction. Lactoferrin or luciferase, these are the luminescent organs. Then electron transport particle, which involves in the mitochondrial energy transducers. This energy transduction, it's activating some pathways. We discussed some pathways in the previous state, and I am not fully covered this oxidative phosphorylation process. I'm seeing some questions which carries this topic, oxidative phosphorylation. So I am included this in this class. In this oxidative phosphorylation, electrons from the reduced cofactor, okay, that reduced cofactor NADH and FADH, this passed to protein in the respiratory chain. This is make a oxidative phosphorylation. That reduced cofactors are the NADH or FADH2. These are passed to protein in the respiratory chain. This is a very important keynote, because whenever they are asking the question, they are asking about the involving compounds. That may be SAFTP, that NADP, NADPH, that all those things, ATP or FD, that kind of questions. In that kind of way, because this is they may ask like a one word question. So we need to understand the concept, main concept. In this concept. They are oxidative phosphorylation, they are using the NADH and FADH2, that is the reduced cofactors. In eukaryotic, oxygen is a limited electron acceptor for these electrons. So in this way, that NADH and FADH2, it's a reduced cofactor which carries the electrons. These electrons are the ultimate that are transferred to the oxygen. That oxygen is a acceptor. Okay, that ultimate acceptor. So they are added to the that oxygen, that receiving the oxygen. So energy of oxidation is used for the phosphorylate FD ADP. This energy, so there is a that electron and oxygen binding make a energy, that energy helpful for the ADP phosphorylation. This is the overall thing which involved in the oxidative phosphorylation pathway. Here it's the simplest form. It's very simplest because NADH2, NADH and FADH2 is that's releasing the electron. That electrons transport chain, which makes the utilizing the oxygen, because that is the acceptor, and this make sense of it, that oxidation of phosphorylated ADP. That ADP plus PA, it's make a ATP, that is a phosphorylation process. It's a elaborate pathway. In this first step they mentioned that, right, that electron transport chain. In this electron transport chain, which they are making utilizing this NADH and producing some electron H plus ions. This produced H plus ions are directly added to this pathway and producing the ADP to ATP. So here 30 to 34 ATPs are produced at the end of this reaction. The very notable thing in this topic, here one NADH, it gives 3 ATP. Okay, so this is the example. Here one NADH is utilized in this electron transport chain, that helps to produce at the end it's a 1 glucose yield, that and its yield helps to yield of 30 to 34 ATP. So how much amount of the NADH are utilized? So 10 NADH utilized in this process, that producing 30 ATP. Likewise here FADH2 they are utilizing, that here you can see here one FADH2 it's make a yield of 2 ATP. One NADH it helps to 3 ATP production. One FADH2 helps to 2 ATP production. This is very notable thing in this oxidative phosphorylation, that make a questions with this ATP that product and yield. Once again tell, one NADH yields 3 ATP, one FADH2 its yields is 2 ATP. At the end of this phosphorylation cycle, oxidative phosphorylation, that are producing 30 to 34 amino acids with 1 glucose. Okay, then the coupled reaction. This coupled reaction is also another part of the energy transduction process, that is the bioenergetics. In this way, the coupled reaction, that called the energy is coupling between two reactions. The cell make a endergonic reaction happen by supplying them with free energy released by exergonic reaction. This normally pathway, the exergonic in the path 1, internal cell make a utilizing the producing some energy. That energy is utilized by the transfer of energy from one reaction to another order of second reaction. So one reaction is finished in one pathway, that producing some energy, that energy utilized by for next pathway. Here you can see the glucose is converted into here the glucose is converted into CO2 plus H2 plus heat. That this is the glucose metabolism, that is the exogenic reaction. Okay, that is the exogenic reaction, the glucose releasing the CO2 H2 plus heat, that produced that ATPs. In this reaction we are releasing ATP. This ATP is utilized by protein amino acids. That amino acids are synthesizing the protein. Okay, so that is the endergonic reaction, the protein synthesis pathway. That glucose is degraded into the carbon dioxide, H2 and heat. During that time that are releasing the ATP. The ATPs are utilized in the lipid pathway to produce the protein synthesis. Okay, and then it's a protein metabolism pathway is called as the endergonic. The glucose metabolism pathway is exogonic, and protein synthesis pathway is endergonic. And a very important topic in the protein concept is the Ramachandran plot concept. In the exam point of view, it's very important one. So I am included this in this class because and that during that class time I am not explained the Ramachandran plot concept. It's very important. In this Ramachandran plot diagram, we are having the that phi and psi, that psi value plots are here. Okay, here that x, okay, x origin having the phi value, y origin having the psi value. That is, I am telling the basic things, but it's all our things are very important, because in that exam they are providing the para of question with this basic information story. In that we want to understand the concept and answer it. So only I am telling the x axis carries the phi values, that pi values, then that y axis carries the psi value. In this we have the 0 at the middle. Okay, here this is minus phi, this is a plus phi. It's the likewise here minus phi, psi and plus psi. Here in the exam they provided the shaded area with the yellow color area, that is a white area, because that is if there is a computer based exam, they may provide us a color one, or they may given us a black and white. In the exam there it's a dark and it's a white area, that light shaded area. Okay, here I am explained with this color, and then I am explaining that black and white image. This white region basically disallowed for all amino acid except glycine. Okay, except glycine, it's disallowed. It's not allowing the amino acids. So if there is any amino acids are there, that won't allow. Then red color region. Okay, this red color region that allow the regions, namely alpha helix and beta sheets. Red color region, it allows only alpha helix and beta sheets. Then the yellow area is the outer limits. Outer limits means except the alpha sheet and beta helix, in the protein that are many amino acids are present in the loops. Many amino acids are there that are all be present in that yellow color region. In this, the beta sheets count is high. In a normal, that beta sheets are high, rich in the amino that proteins. We all know the protein is majorly is made up of amino that beta sheets. Some proteins are alone having the alpha helix. that most of the that carries the beta sheets and alpha helix, both are present in it. So this beta sheets, if the psi plus values, you can see here psi and phi minus, this region carries more beta sheets, means that makes some sense in the Ramachandran plot explanation. Here they provided the example, that image alone we need to describe. Okay, in this image that beta sheets counts are high, that left-handed helix in this phi region, this region for this this minus, both are minus, phi and psi both are minus in the left-handed alpha helix region. Here based on this quantity they are provided the number, so we can tell that protein carries the left-handed alpha helix. Here in this A region it's right-handed, here this is left-handed alpha helix. So here this is a small one, that is a small quantity of left-handed alpha helix. In this so in this image itself we can understand the plus psi and minus phi is beta sheets, minus psi and minus phi carries the right-handed alpha helix, plus psi and plus phi that carries the left-handed alpha helix. So the empty region, its white region, is except glycine. If here you can find any dot, means that protein carries glycine. How much amount of glycine carries? So glycines are present in the minus that last region. That maybe glycines is also present in this area also. The empty region of there is no representation of alpha beats and zeta alpha helix and beta sheets, that is a region called the minus minus psi and plus phi. The very important thing in this Ramachandran plot, the plus psi and minus phi region is for the beta sheets, and minus phi and minus psi, this region is for right-handed helix, alpha helix, and plus psi and plus phi for left-handed helix. That's all. The all about thing, the Ramachandran plot is very important. So they are provided the para-like question, but that is very too easy when you understand the concept of the Ramachandran plot. Okay, here as I'm mentioning already, here the for understanding this type of questions, okay, the steric clashes, that is the additional interaction to stabilize the structure. So there was some additional compounds are there that may be functional, that play as functional significance. Amino acids, it's conserved within the protein family. So some specific protein families carries the some amino acids. Okay, here in this image itself we can see the cumulative dots, and here you can see the cumulative dots. They are provided that this image alone. So you can see this region carries the alpha helixes. The region carries the beta sheets, that is the rich in the alpha helix and right-handed alpha helix. There are small quantity of left-handed alpha helix also there. Some white regions carries the small dots, that may be the glycines. Okay, in that that beta sheets antiparallel, parallel also, right? Likewise, like this alpha having the right-handed, left-handed, right? So similarly, this beta sheets also having the parallel anti-parallel. Okay, in that image, the internal red color region is there, right? This red color region we having the in the center portion and external portion. The center portion is that 3 points here. You want to note that this region, okay, lower region means the parallel beta sheets. Then this intermediate region, it's represent the anti-parallel sheet. The right twisted beta sheets are in this top. This center area. Then collagen triple helix also there, that is occupying the top area. This are all about this Ramachandran plot. Okay, this I think you may understand these concepts. The amino acids are dotted in that area, though specifically that amino acids are placed in the beta sheets, or that may be placed in the alpha helixes. So we need to understand the basics of this concept. Then next remaining topic is a immunodeficiency, and in that classes are not covered. Here I am going to explain. In this immunodeficiency, we have two types. One is a primary immunodeficiency, another one is a secondary immunodeficiency. In the primary, it's a predominant antibody defect. It's combined with a T cell and B cell defect. Cellular, it is also called as the cellular immunodeficiency. It's a complement defect, it's a phagocytosis defect, disease of immunodysregulation. In this secondary, that systematic disorder, there is a systematic disorder, and immunosuppressive one, that may be cytotoxic, that is cytotoxic chemotherapy due to the cytotoxicity. So some that cancer patients have lost their hair, that is also the some immunological response. That is maybe the radiotherapy, that bone transplantation makes some side effects. So that are all the secondary immunodeficiency. One is which cause the directly affect the our pathway, means that makes sense of that primary immunodeficiency. If we are affected by the secondary thing, that may be a drug, radiotherapy, or anything. It's external factor which is directly affecting external factor, that activating one pathway, the activation of the particular pathway mechanism that dysregulate the one another pathway, that makes some immunological response. That immunological response called as the immunodeficiency. And this is a very important, because I am also facing this kind of questions in the CSIR examinations. I am refer that questions, they are provided like a table of that match the following question, and they provided as a 2 mark and 1 mark questions. Here this table is very wonderful and explanatory one. Here T cell disorder, okay, this kind of once the immune system affecting the T cell, that T cell disorder is having the 4 different types. That is the severe combined immunodeficiency, the Wiskott-Aldrich syndrome, that is one called of the immunological effect, the atossia telangiectasia, and DiGeorge anomaly. These are all the diseases which are caused by the T cell disorder. If B cell defect makes, if due to the immunological response, the B cells are defected, that may be the this agomaglobulinemia, and common variable immunodeficiencies called selective AGA deficiency, AR agamaglobulinemia, and hyper IgM syndrome XOL. These things that happen. It's very important. In that it's that word itself you can understand. The B cells mean that affect the, that depends on this that IgGs. So you can, in this word itself you can understand and memorize the concept. And phagocytosis based disorder, it makes a chronic that granulomocyte disorder, leukocyte adhesions. Now all those things are, it's related to the our immunological response. Okay. And that cyclic neurotropionia, these are all things that happen. And there's complement disorders. These are all the complement disorders, that factors defections and propyridine deficiency. These are all things are there. In this secondary metabolism, we want secondary deficiency, immunodeficiency. The main reason for the developments are the HIV based one. The second thing is the HIV is developed with the acute immune disease. That is the secondary thing, in which is caused by the secondary thing. It's not caused by the direct virus. The viral system affect the cell. The cell system make a sense to that made activating the human. Then ionizing the radiation like that. Then it's protect, and unreasonable treatments are called the cytostatic immunodepressant. These are all the drugs, hormones are make that are affecting the cells. That cells are activating the immunological response. That immunological response are make that are activating the pathway of immunodeficiency. In this immunodeficiency, we are having two different type. That two different type called as the congenital and acquired immunodisease. In this congenital immunodeficiency, it's a failure of T cell development. That is the severe combined immunodeficiency. Okay, that is a very, very important thing. A severe combined immunodeficiency is a congenital immunodeficiency. And if the failure of B cell development means, that is the X-linked agammaglobulinemia. Then function of P cell and T cell both are affected at a time, same time, means that makes a different type, different diseases. The main thing, if the T cell alone the D cell development alone affected, that alone the affected by the immunological response, that make a severe combined immunodeficiency, the SCID. If the B cell development alone affected, that means that produce the X-linked agammaglobulinemia and globulinemia, and both function of B cell and T cell both are combinedly affected, means that producing these diseases. That is called X-linked hyper IgM, that's X-linked lymphoproliferative disease, that common variable immunodeficiency and autoimmunodeficiency. These are all the functions. Here I mentioned them. B cell affected means that is the affect called X-linked agammaglobulinemia. In this, that makes some functional defect in the reduce the B cell count. If there is a disease called Ig heavy chain deletion, means that makes a IgG, all those things are absent. So the absence of IgA and IgE, it's make a Ig heavy chain deletion. These are all very, very important things in the exam point of view, because they ask that question, that simple words that are very, very basic question, but due to the, because we are in diverse field, that we are not focusing this kind of things, so we want to understand. If there is a X-linked agammaglobulinemia, means that is the block in a maturation beyond that pre-B cell. It's mutation in the B cell tyrosine kinase. If there is a B cell tyrosine kinase, if there is a mutation in a B cell tyrosine kinase that producing the that conjunctival immunodeficiency, that so that in that help that make a B cell immunodeficiency. That very very important thing. And chromosomal deletion at 14q32 chromosomal deletion, it's make a Ig heavy chain deletion disease, that is a congenital immunodeficiency. That T cell immunodeficiency, that is called the George syndrome. In this, D cell is decreased, the serum Ig also, immunoglobulin level also decrease. In the anonymous development of third and fourth branchial pouch leading the thymic hypoplasia, it's a T cell immunodeficiency mechanism. Okay, this is the basic thing which is very very important in the exam point of view. An acquired immunodeficiency. In this acquired immunodeficiency, we have it's called as the secondary thing, as I mentioned in the earlier style. That is acquired immunodeficiency symptom. It's caused by the HIV virus. It's a statical one. It's a long time one. It's major type. It's cancer, bone marrow, and those are particularly affected at the bone marrow level and blood cells level. In this also we are having some diseases. In human immunodeficiency virus, that is HIV, it's depletion, that affects the depletion of CD4. It affects the depletion of CD4 helper T cells. Then irritation and chemotherapy treatment of cancer, it's decrease the bone marrow precursors for all leukocytes. Then involvement of bone marrow by cancer, that is the metastasis and leukemias, that is also the secondary and acquired immunodeficiency. That are reduce the site of leukocytes development. And protein calories malnutrition is also the disease. It's metabolic. It's affected due to the metabolic rearrangement inhibit lymphocyte maturation. The metabolic rearrangements that derangements are the inhibit the lymphocytes maturation. And removal of spleen, it may also cause the immunodeficiency. It's decrease the phagocytosis of microbes, that is another deficiency, that is the acquired immunodeficiency. These are all the sequential process in the acquired immunodeficiency, that is the AIDS. In that way we having the different. First is a human immunodeficiency virus make this thing, or it may think a chemotherapy effect. It may be the bone marrow based cancer, involvement of bone marrow by cancers as a metastasis or leukemias. And protein calories malnutrition also make a immunodeficiency, and removal of spleen also make a immunodeficiency. And the next point of view is very very interesting. It's aging. In the aging mechanism, first we are all thinking how it is that aging makes. Okay, it starts from the molecular, the whole body, that whole bodies are producing the some level of energy to organs, that organs are producing some level that are activated by the cellular level, the cellulars are activated by the molecules, that molecules are activated by the submolecules. In that same kind, it can use entropy increase, that energy. Okay, that the entropy released, the energy release is increased, that functional groups or cross links are making higher. So there is a free radical and diapatase glycogens are there. So this free radicals, diapates and glycogen that activates the aging gene and telomeres. Due to the activation of aging gene and telomeres, that high flick limit of cell proliferation, it starts. So it stops. That is a flick. That is a cell proliferation. In this stem cell lysosomal mitochondria are the also on the it's they're activating their pathway, and then that activating the brain aging, atherosclerosis and endocrine. So the body size become reduced after some particular age. Then that caloric restrictions also there. These are all the aging mechanism. Here that's aging is proteins which involves in the, here this is a just explanation what is the process happen in the human body. In this, proteins are involved in this aging process. Okay, in this aging process, the first thing, the activation of the this aging, it leads to hypertension. All pathways are interlinked, that in that pathway the aging, it's low expression of SIRT protein and it's a high expression of FOXO protein. So cell cycle regulations are it's affected. Then that MMP protein and TIMP proteins are activated, and ion channels also activated. The mitochondria, all those things. If the reduction of SART protein and high production of high activation of FOXO protein, that makes the apoptosis, autophagy, that proliferations. So vascular modeling happen. This the vascular modeling, it's make a hypertension. Then cell cycle activation, where is it? Cell cycle activation, it makes the fibrosis process and stiffness, it's a higher stiffness. So after particular aging, the stiffness become reduced and it makes the elasticities. That is a vascular that classification. Then the ions, that is a mineralization, it's also in the vascular calcification. Then the calcification, it's make a hypertension. Then mitochondrial dysfunction and NOx activation, and the ROX higher and the nitrous oxide has become low. That makes a low height, the vasorelaxation and high vasoreactivity, that are reducing the endothelial dysfunction, that are affect the endothelial dysfunction. Then inflammatory genes called MAPK, that MAPK, it's involves in the that activating the pathway called cytokines and chemokines, that are VCCAM1 and VICAM1, and that are activating the vascular inflammation. All those things are vascularity pathway that make a hypertension. So this is only one pathway explanation. Okay, with this aging, which is makes due to the it's another topic apart from aging. This is another important thing, because the apoptosis pathway is a programmed cell death, which is very 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 important in the human cell development. If there is no the programmed cell death happen in a cell in a human body, that make a cancer. So the faculty enzymes must be digest the that cells. That cells are that's the scavengers cell are, the two ways are here. This is the automatic faculty enzymes degradation or scavenger cells, that some cells are engulf the cells and degrade it. Okay, which pathway is involved in the apoptosis? It's a very very important thing. Here the proteins are called endocrine proteins are here. These proteins are receiving the signal for that FAS protein receiving the signal from the extracellular membrane, that activating the pathway called caspase 8. The caspase protein, which is a major protein involved in the apoptosis pathway, what's this is a basic thing which we want to remember, because whenever we are going to, they are asking the question like the apoptosis, that they are included the word called caspase. This is a default one. So this that caspase 8 activation, it's activating the T-bit protein on the mitochondria. So now process is start with mitochondria. So mitochondria activating the pathway called SMAG diablo, and it's activating the pathway called IAP. That IAP is activating the X castioner caspase, that is the caspase 3 and caspase 7 is also important. The execution caspase is a combined form. That combined form is make a cell death. Okay, another pathway is the mitochondria activating the cytochrome C and cytokine C, or that APAF1 caspase 3. These combinations are activating the cell death. Another thing, the endoplasmic reticulum having the caspase 12. This caspase 12 added at the level of this executioner caspase the complex form. That complex form make a cell death. Here I am included how the Drosophila getting the apoptosis process, and mammal cells are how they are activating the apoptosis. In this Drosophila, that are the RHC family, that families are that having the HEAT, GRIM and sickle rappers. These are all the genes which are activating the pathway called DRP1, that dranic dark. These are all the which proteins, which genes involved in that pathway. The effector caspase are activating the cell death path, the apoptosis pathway. That may be activated by the junk pathway. If there is a junk pathway activation, so there is no apoptosis cell death. Very very important thing that you know, understand. If there is a junk pathway activation happened, means there is no apoptosis in Drosophila. Then in the mammalian cell system, that extrinsic pathway and intrinsic pathway. In the intrinsic signal, that IAP inhibitors are activating the caspase 9. Okay, so that this you can see the IAP, this activating the caspase 9, that activating the caspase 3 or 7, that is the effect of the executions protein. Here the executions are the caspase 3 and 7 in a human. That's caspase 3 and 7 is the executors. Here in the Drosophila, DRCE and TCP1 is the transducer. If the caspase 3 and 7 complex makes sense of activation of apoptosis. So in the intrinsic pathway, BH3 only activated the box and BCL2. This box and BCL2 activation of the cytokine C, that is the complex with the APF1. So these combinations are activating the IPA pathway, that activating the caspase 9, caspase 9 activating the caspase 3 and 7. Okay, next the remaining topics I will discuss in the time taken. Some important topics, not too much cover all those things. This is just a beginning. It's just a starting point. After this tomorrow class, every day please study the concepts. Take your notes and take the small notes, because in this CSIR we can't expect the focus, because this for example in this apoptosis concept also, in this apoptosis, in this current period, many research are there. I am explaining just the basic principle. In the exam point of view, they may ask with the drug. Okay, this drug activating this pathway, what happened, or that is the questions. If that drug inhibiting the caspase 9 means that activating the that stop the apoptosis process, so that cancer may cause. So these are all the things are very very important. So in this uh, this apoptosis, that necropoietic process also happen. This is that is a the necroptosis. If there is a inhibition of MLKL protein means that stop the necroptosis process. These are only for example questions that in the exam point of view they may ask with the latest research point of view. So you want to refer some extra new articles based question. If you are taken any topics means just understand the latest concept, because in the exam they are providing the in the exam they are not providing the principle in definition. They are asking the internal topic question. For that we want to understand the latest research which is going in the laboratory level. So understand the concept and correlate with the research point of view, we can get the higher score in a CSIR. Tomorrow we will cover the remaining some important topics. Apart from this important topics, please refer some notes, books separately. The book link also I am included, that I am loaded in the Google Classroom and your mail ID. Please refer that. Thank you everyone. If you have any queries, please post it in chat box. Yeah, sure, I will upload the previous year question. Actually at the end of the section I am having the plan to upload. Today I will make that. Yeah, cell death is very very used because if there is no cell death happen in a human cell, that is, so there is a accumulation of cell in a particular place, that is called as the cancer. That is the not only the uncontrolled proliferation. So if there is a normal control but there is no cell death. For example, you may know the in Tamil the Kalani, that is the one pain is there. There is no uncontrollable growth, but there is no cell death. So the cells are accumulated in particular place, there is a no removal, so that make a pain at the place. So cell death is very very important for all the human and animals also. Cell death is very very important. After reaching that's why only they mention the aging, that cell death that develop their organs, develop their body, that new cell generation only activate our new pathways. One thing I want to let you know in this class, if you are I am biotechnology, I am not aware of zoology and botany. Okay, if you are a zoologist, you may not aware of, you are not familiar with the genetic engineering, or you may not aware that you are not well versed in the topic cell biology, that basics things. If you are in microbiology, you are well versed in cell biology, microbiology, that concepts, but you are lagging in the botany. Okay, in that way, please take the 12th standard book. Okay, 12th standard and 11th standard NCERT book, that gives lots and lots of basic ideas. Okay, it's very important. I am feel good. Okay, they are in that 11th and 12th NCERT book. They are, because we as a student we can't cover the whole book. If you are a reader, that may be the possible one, but as a student, the full book reading is uncomfortable one, and it's not easy. So for your exam preparation, for your basic understanding about the genetic engineering, about the basic concept, please refer the 12th and 11th standard NCERT book. I'll also send the link when I was uploading when I will uploading this previous year question paper. I'm also include some websites also. Some websites carries the previous year question papers, previous year books, the subject books. So I'm included that websites in the Google Classroom, and the description also. In the next tomorrow I will make that. In the description I will upload, because tomorrow is the last day. You may whenever you want to refer this content, please go through the video, and that description also. The description I am included the website, some websites, and that book links, that which will helpful to you. So thank you everyone. Thank you for your patience.